Good afternoon, everyone. It's my absolute pleasure to open the first Alumni Lunchtime webinar presented by esteemed immunologist, researcher and alumnus, Professor Greg Woods. This webinar will form a living record of university achievement, which you'll be able to access from our website. For the past 10 years, Greg has been leading a team who are investigating devil facial tumour disease, and this work has shown remarkable progress in understanding the immune system of the Tassie devil and how cancer escapes immune recognition. Over this period, Greg's been awarded more than $2 million worth of research funding. He's also published over 90 peer-reviewed scientific papers and has supervised approximately 20 PhD students to completion. As a UTAS alumnus myself, I never suspected when undertaking an arts degree in the early 90s I would be managing the central fundraiser for the Tassie Devil, the Save the Tasmanian Devil Appeal, which is administered through the University Foundation. And I certainly never imagined I'd be introducing a professor whose work centres on transmissible cancer. I remember when I started in my current role, meeting Greg was a real and first priority, and I recall trying to look knowledgeable while he explained his work. This was not as challenging as I first imagined, for Greg has a real genius for making complex topics both interesting and engaging, and this is why today's webinar will be such a treat for listeners. Greg's work is at the cutting edge of science. Over the last 10 years, he and his team have rekindled hopes of saving our iconic animal, and the implications and importance of their work should not be underestimated. Following Greg's formal presentation, we will allow some time for questions from listeners. So please now sit back, listen, learn and post questions via the chat box underneath the presentation on your screen. Please note you'll need to be logged into live stream to use the chat facility. All of your questions will be visible to other listeners, and due to time constraints, we will just choose a selection. And now, Professor Woods.
Thank you, Rebecca, for that introduction, and welcome everyone to whether you're listening live or whether you've decided to download it at some convenient time. I'm an immunologist and I currently work at the Menzies Institute for Medical Research. Now, Menzies is basically looking at human cancers. And why is a person working on devils in Menzies? Well, it's a long story which I haven't got time to go into. But basically, cancers are the same whether they be in animals, devils, or humans. So I'm going to talk about some of the research that we've been doing on the immunology of devil facial tumour disease. I'll give you a bit of an overview. There's a website here, www.tassiedevil.com.au, which gives you a lot of good background information, and I'm sure a lot of you have logged into that already. I'm going to mention 5 names: Rod Wolf, an anonymous Northcote man, Peter McAfee, Edward Jenner, and Frank Fenner. Hopefully some of you are scratching your head and thinking, what are these names? You may have heard of one of them, some of you may have heard of 2, but I think I'm the only person that knows all these 5 names. The mystery of these names will be revealed later on. The science, I'll talk about a bit about the cancer, or the cancers, their origin, or its origin, and their distribution. As an immunologist, I'll talk about a bit about the immunology, very briefly, some of our vaccine research, and future plans, because I believe we're on the possibility of making some major inroads to protecting the Tasmanian devil from devil facial tumour disease. The cancer. In 1996, in the far northeast of Tasmania, you can see a little grey area and a red star. That's the very first case of devil facial tumour disease. It happened in one particular devil. And for the next 10 years, the tumour spread across half of Tasmania. And you can see that grey shadow is where the tumour is spreading, and in its wake it has been killing devils, those red stars. It's been killing devils and continued on for another 10 years, 2014. I've chosen 2014 here. The black dark shadow has covered most of the state, and 2014 is also an important year, and that will become relevant a little bit later on. So the disease started in the far northeast and has spread across most of Tasmania. There is a disease-free population in the far northwest of Tasmania, but the rest of Tasmania, the devils have been affected by devil facial tumour disease. So, sorry, there's a horrible picture. Devil facial tumour disease is a transmissible cancer. It's called devil facial tumour disease because the tumours occur on the face, and the top 3 pictures will show you some horrific tumours of devils that have been caught in the wild with these massive tumours. The bottom 2 pictures, the one on the left shows a horrible cancer on the snout, but if you look at the bottom of that, in the bottom jaw, you can actually see some tumour, and the tumour has taken out those bottom canines. So it can also occur inside the oral cavity, and that poor dead devil on the right shows you how weak it's looking. Devils that get infected by devil facial tumour disease die from this disease. We have recent evidence that maybe a few devils may be resistant, but 99% of devils get killed by this particular disease. It is a true cancer in that it kills its host. So what is it? Devil facial tumour disease is a cancer, as I said, but it's a cancer of Schwann cells. Now, work we did with Elizabeth Murchison when she was at the Sanger Institute, and now she's at the University of Cambridge, using some genetics and sequencing, discovered that the DFTD is a cancer of Schwann cells. Now, Schwann cells are those cells that coat peripheral nerves. Peripheral nerves you'll find in the periphery of the body, and in the devils, the face has a lot of peripheral nerves because that's where they use most of their senses. They use their whiskers to feel and sense their environment. So it's a very good environment for peripheral nerves to grow, and Schwann cells. So those 3 pictures on the left under the title called H&E just shows a basic picture of the tumour. And there's 2 squares. One on the left is a peripheral nerve, and one on the right is the tumour. So devil facial tumour disease originated from Schwann cells. But it originated, and I still can't believe this because it is just too incredible to be true, but it is. It arose from one single cell in one female devil sometime prior to 1996. So devil facial tumour disease arose in one cell and has spread for 20 years throughout the whole state, through most of the devil population. Suza Tovar, postdoc now in my lab, discovered that a marker called periaxin, which is on that right column, is a good way in which we can identify devil facial tumour disease cells. Periaxin gives us a good diagnostic marker, and this marker will also be important later on in 2014. So devil facial tumour disease can be identified by the expression of periaxin, is a cancer of Schwann cells, and it's been spread through the devil population. So we have a dilemma. How can devil facial tumour disease be a transmissible or a transplantable cancer? When cells go from one person to another, the immune system is really powerful and rejects the cancer cells, rejects the tumour cells, or rejects the transplant. But this doesn't happen with devil facial tumour disease. So our first mission was to try to understand how the tumour cells, devil facial tumour disease cells, can go from one devil to another without being rejected. We know that devils bite each other, and that picture on the right-hand side just shows devils playing, but we know that devils will bite each other on the face, and when they do bite, they will transmit a few cancer cells. Now remember I said the face has a lot of areas where the peripheral nerves can grow, so it's a nice environment for the tumour cells to grow. So a tumour cell will go from one devil to another in the face, the tumour will grow, and then the process will occur again, and that devil will bite another devil, and it will continue. So the biting behaviour accounts for the way in which the tumour cells can be transmitted. But the big question is, why are these cells not immunologically rejected as an allograft? In other words, why doesn't the immune system see these tumour cells from the other devil as being foreign and reject them? The 2 questions can be answered by looking at whether it's the devil, is the immune system of the devil something wrong with it, or is there something special about the cancer cells? Our first role was to look at the immune system of the devil. So we had to analyse the immune response of Tasmanian devils, and the best way to do that is getting a blood sample. So devils, like the rest of us, have a good blood supply, and you can analyse a lot of immune responses within the blood. When you analyse the immune response of humans, you just need a blood sample. So basically what we did was we trapped our devils. So we had devils in captivity, so we trapped them, And because devils, well, you want to make sure they're nice and quiet, we had to put them into a sack. And once we're in the sack, we anaesthetise the devils, and then we could collect the blood. Now, interestingly, devils in the wild are really calm. You can pick them up and bleed them. Once they're in captivity, yes, they're not as calm, and you have to anaesthetise them before you can take a blood sample. So we would trap the devils, anaesthetise them, and to get the blood, we'd have to use the jugular vein to obtain the blood sample from. And this picture shows a devil that's been anaesthetised, and we're collecting blood from the jugular vein. Takes a bit of experience, and our researchers and our vets are very experienced in collecting blood samples, and the devils can be bled quite happily, and they'll return back to their captivity without too many problems. So we'll obtain the blood sample, then we'll take it to the laboratory to analyse. And because our laboratory is a PC2 laboratory, you need to dress up in lab coats normally. And this shows you a Tas Devil, who's one of our mascots. He was going to go into the lab to show us, but I'll have to show you with some of our research staff. Into the lab, it's a PC2 lab, so it has special requirements. And in the lab, we can analyse the blood sample. Now this picture shows 2 blood samples, which could be from a human, but in this case it's from Tasmanian devils. In that blood sample, we have red blood cells, which is why it's red. We also have white blood cells, and we also have the serum. We use the white blood cells to analyse the immune response, and the serum to look for antibodies. So the blood is a very important resource for analysing the immune response. So we've done a lot of work. We've taken blood, looked at different cell populations within the blood, immunised animals, devils with different antigens to look at the immune response. And to summarise the work, we would first have to collect the blood, as we talked about, separate the blood, so take them into the laboratory. And remember I said we have red blood cells, white blood cells and serum. We want the red cells, we want the white cells, and we want the serum. The white cells and the serum we use to analyse the immune response. The red blood cells we don't use for analysing the response. So collect the white cells, particularly the lymphocytes, and the serum to analyse the antibodies. And we've done numerous tests over the last 10 years. And to summarise that in a single sentence, Tasmanian devils have a very competent immune system. Every test that we've done shows that the immune system of the Tasmanian devil is fully effective. So a suppressed immune system does not explain susceptibility to the transmissible cancer. If we immunise devils with antigens, they produce a response. If we look at their blood, they have all the cells that are required, so they have a good immune system. So that takes us back to the tumour cells. Maybe there's something special about the tumour cells that prevents an immune response from happening. So we know the devil's fine, but the cancer, it's somehow avoiding the immune escape. We initially thought that devils were genetically too similar, and so there was a lack of genetic diversity that prevented the immune response from being activated. That's part of the answer, but it's not the main answer, because we did some basic experiments where we did a skin graft. We grafted the skin from one devil onto another devil, and it got rejected very readily. So it tells us there is sufficient genetic diversity amongst the devil population to recognise foreign cells as being foreign and induce an immune response. So lack of genetic diversity is not the main answer, it may contribute. What we had to do was to look at the immune response. And in this diagram, we have periaxin on the left. Now you remember periaxin is the marker that we use to identify devil facial tumour disease cells. And on the left column there, under the periaxin, you'll see some blue cells and some brown cells. Those brown cells are the tumour cells, and the blue cells are just normal tissues. Then the next 3 columns, we have CD3, CD4 and CD8. They represent the immune response. CD3, CD4 and CD8 represents different populations of T cells. And if you look carefully, you'll maybe see a couple of CD3 cells, which are the brown cells, very few CD4 and very few CD8. If you look even more carefully, you notice that where the CD3 cells are, it doesn't correlate to the tumour. So what we've discovered here, and it's been repeated many times, is that the tumours will grow and there's no infiltration of T cells or the immune system. So basically what's happening, there's a lack of immune cell infiltration, which means the tumour is growing without alerting the immune system. It's growing and the immune system doesn't know it's there. So it's just avoided completely. So we have to ask, why are these tumour cells not recognised by the devil's immune system? Because they should be. Allergenic responses are really powerful. So then we looked at the MHC gene expression, and this was done in collaboration with Hannah Siddle, who was at the University of Cambridge and now is at the University of Southampton. So MHC stands for the Major Histocompatibility Complex, and I liken that to the flag on particular cells. So it's a marker that the immune system uses to identify cells as belonging to self. We all have flags on our cells that tell us that these cells belong to self. The flag of someone other cells tells us it's foreign. If the flag is wrong, i.e. from another cell, from another person, you should get an immune response. Now down the left hand we've got cell lines, which are cells we've grown in culture, and down the right hand under that bracket we have DFTD biopsies and host spleen. These are primary tumours. Without going into the details, what we have found is that the genes that code for MHC, the genes that code for immune recognition, are not expressed on the DFTD cells. So the DFTD cells don't express MHC genes, so they don't alert an immune response. So it's not lack of genetic diversity, or that may contribute, it's the fact that they don't express these genes. The cells are basically invisible. And I can liken that to the fact that DFTD cancer cells avoid immune recognition because they do not express MHC. The devil cancer cells are invisible to the devil's immune system. Some of you may be Harry Potter fans and be familiar with the cloak of invisibility. And we've always wanted to have a cloak to make us invisible. And that's what I liken this to the devil facial tumour diseases. They are invisible to the devil's immune system. The tumour cells will grow without alerting the immune system. They can just grow bigger and bigger and bigger, and then they can kill the devil. Now, devil facial tumour disease is a true cancer because it will metastasise and you get lots of cell division. So, is there any hope? Maybe DFTD cells are antigenically inert. So the next thing was we thought, well, if they don't express these MHC genes, they can't be seen by the devil's immune system. Can we turn them back on? When we looked at the DNA of the devil tumour cells, we were very pleased to find that those genes are still present. So they haven't been lost, they are still there, they're just turned off. So then we looked for the light switch to turn these genes back on, to make them become visible again. But before we did that, we wanted to see whether or not we could induce an immune response. So we immunised devils with population of devil facial tumour disease cells. So we grew the cells in culture, sonicated them, added some adjuvants, which are basically stimulants of the immune system, and then injected them into devils and analysed the response. So these next three slides very quickly show sonicated, frozen thawed, and irradiated cells. When these cells, different populations, were injected into Tasmanian devils, they induced an immune response. So this provided good evidence that the immune system of the Tasmanian devil can recognise DFT tumour cells under the right conditions. So those right conditions required large number of cells and adjuvant. We need to improve this, so we started looking for that light switch to turn those immune response genes back on. So DFTD cells do not express MHC, but if they're treated, and they were treated with a cytokine. Now, we got this idea because we know that when you have an infection, your immune system produces cytokines. And one of those cytokines is called interferon gamma. And interferon gamma basically acts on the immune system to make the immune system be more effective, and actually acts on the target cells to make them more antigenic or increase the expression of MHC. Now that diagram at the moment, MHC1, TAP1, TAP2, PSMB8, shows the genes expressed. The MHC1's there, but TAP1, TAP2 and PSMB are not present. Without those latter three, the immune system won't function. So we treated those cells with interferon gamma, a cytokine, and the light switch came on. You can see under the T, the treated, that bright white light. We have turned the light switch back on, and that column in the right shows what happens on the surface of the cell. So DFTD cells exposed to interferon gamma re-express their MHC, and that should make them antigenic. So the tumour cells are modified and they will become visible to the immune system. So this is an improvement on the previous, where we just injected straight DFT cells. If we could alter them to express the MHC, we want to determine whether that would produce a better response. So the next was a trial on 4 devils: Madim, Badger, Stinky and Tip. What we did with these 4 devils is that we treated the cancer cells, the DFT tumour cells, in the laboratory to upregulate MHC and immunise these 4 devils. Now we are limited with the number of devils that we have, so we did a trial of 4 devils, and the next slide shows we divide them into 2 groups, 2 groups of 2, not very many, but the first one we injected with MHC positive sonicated cells, so these are the tumour cells, treated in the laboratory with interferon gamma, and then to express MHC, then they were sonicated. At week 0 and week 4, we gave 10 to the 7 cells. Then week 8 and week 12, we injected these devils with irradiated cells. The second group of devils, we tried to improve this by using irradiated cells. So the DFTD cells were treated with interferon gamma, and then rather than sonicate them to smash them up with ultrasound, they were irradiated to keep the cells intact, and they were injected with irradiated cells at week 0, week 2, week 4 and week 8, and we analysed the response. So this is a diagram of a flow cytometry plot just to show the response, and what we discovered was that the devils immunised firstly with the sonicated cells produced a better response than the devils that were first immunised with the irradiated cells. The other thing it shows that all 4 devils produced an immune response, which is a really encouraging response, because in our previous trial we weren't getting every animal to respond. But we needed to increase numbers to make this realistic, to make this real, to be more convincing. Now the Save the Tasmanian Devil program has developed an insurance population, and this insurance population are devils bred in captivity or in free-range enclosures, and their aim is to reintroduce them back into the wild. But the problem is they can't be reintroduced back into the wild whilst their disease is still there, because they'll become susceptible to the disease. Our problem is we haven't got access to enough devils to test our immunisation protocol. So the logical thing was, if we could work together, if we could immunise a large number of devils and release them into the wild, 1, we would get more devils to test, and 2, if that was protected, they could be released into the wild. And that's basically what the next step was. So we developed a schedule, and this rather complicated diagram just shows you what we had to do. That we had 2 lots of devils. 1 were in the Launceston, these were boys, and 1 were found in Vishnu, which were free, girls. We had to immunise these devils on 4 separate occasions at a month apart, and then gave them the booster just before release. So this took about 8 to 9 months to do. So it was a major undertaking to immunise, to bleed, and to analyse response. But at least we increased the numbers. We had a total of 19 devils. And the next slide is that these 19 devils have been immunised. We released them in Narantapoo National Park in September of last year, and we could monitor the progress of these devils. What was really exciting from our perspective was that of the 19 devils that we immunised, 18 of them produced an immune response. Now previously, we had rarely found devils that had produced an immune response. And some of you may remember the great publicity around Cedric about 5-6 years ago, whereas Cedric was the very first devil to show signs of an immune response, and he became pretty much an icon. With this protocol, we have produced an immune response in 18 of the 19 devils. So we actually produced 18 Cedrics in the one go, which is a really encouraging result. So these devils were released in Narantapu, which was probably the world's first trial of a wild vaccine released into the wild, particularly of Tasmanian devils. So they were released. It was a major undertaking, and it was a very exciting time for these devils to be released. But with devil facial tumour disease and the research, we've always found that whenever things are looking good, there's always something to bring you back down to earth. And what happened was that a lot of our devils, 4 of the devils that got released, had been killed by motorists, ended up as roadkill. So the numbers had been reduced, which was devastating for all involved. But there are still devils out there, and we've followed them up at 2, 4, 8, 12 weeks. And in April of this year, 7 of the released devils have been trapped again, and they are still healthy, although it's still too early to tell whether they are protected from devil facial tumour disease. It will take another year to see whether these devils are infected, because the most time for the infections, the transmission to occur, is around about now during the mating season. 3 of these devils have taken up residence in Narantapu National Park, and the rest have dispersed quite broadly. It's amazing how far they do disperse. But this trial gave us some really important background information, and we'll use this for the next trial, which I'll talk about shortly. The other sad thing happened in March 2014. Now, remember when we talked about the spread of the disease, I used 2014. So this is a devil captured on camera on the left in Cygnet, which is in the southern parts of Tasmania, that channel area. This devil looks like it's got devil facial tumour disease. And the picture on the right shows a characteristic tumour which looks like devil facial tumour disease. So our research team, as part of the diagnostic research facilities, went down, trapped the devil, and because of welfare reasons, it had to be euthanised, and we analysed the samples. And to our surprise, it didn't look like devil facial tumour disease. It looked slightly different. And when we looked under the microscope, something didn't seem quite right. First thing was that the tumour cells grew in different sorts of shapes. The second thing was that the tumour cells did not express periaxin. Now, periaxin is the marker that we use to identify devil facial tumour disease. So because the tumour cells look different, they grew different in culture, they didn't express periaxin, we assumed that this was not devil facial tumour disease, although it looked very much like it. We then did through the people in the Royal Harvard Hospital, Karen Dunn and her team analysed the chromosomes, and these are the results that we got. On the top panel, where it says karyotypes normal male on the right, that's a karyotype of a normal male. You've got the 6 autosomal chromosomes, an X chromosome, and a very, very small Y chromosome. That's normal for a male. DFTD, which has been the tumour that was first discovered in 1996, has quite a lot of chromosomal differences, but the devil we discovered from Cygnet doesn't look at all like DFTD. You can see quite differently the chromosomes are not the same. It looks a little bit similar to the normal male, and we know it's a male because it has a Y chromosome. And we thought this was just a spontaneous cancer that arose. Then 6 months later, a second devil turned up, same scenario, trapped, we euthanised, we analysed it. It was not DFTD. It looked different. And then we got a third, and then we got a fourth, and then we discovered that these 4 devils all had a different cancer which was different to DFTD. You can see those bottom 4 chromosomes, those bottom 4 karyotypes, we've got 4 lots of them, and they are all identical. They are all different to DFTD, and they're all different to the normal male. So we have now discovered a second transmissible cancer in Tasmanian devils. because we discovered a second one, we sort of had to rename the tumour. So DFTD, we now refer to devil facial tumour disease. DFT1 is the first cancer that was discovered in 1996, and now this new cancer, this second transmissible cancer, is called DFT2. They are similar, but they're not identical. We still don't know what the cell of DFT2 is, which we're currently trying to determine. As of today, April 2016, we have detected 11 cases, and interesting, 9 of them are male and 2 of them are female. All have been found in the channel area. So two cancers. Is this coincidence or this fate? And this is where one of the mysteries that I mentioned earlier, Rod Wolf, the anonymous Northcote man, and Peter McAfee come in. Rod Wolf was struck by lightning twice, 18 years apart, as a child, as an adult. He was struck by lightning twice. What are the odds of being struck by lightning twice? The odds are very high. So did he have a reason, or was it coincidence? Obviously this was coincidence. So Rod Wolf, also known as Lightning Rod, was struck by lightning twice over a period of 18 years. An anonymous man for Northcote, and I assume he's anonymous because he won Tats Lotto not once, but twice, and won it within 2 weeks. Again, that's a coincidence. So these rare coincidences can occur. Now, if anyone out there does win Tats Lotto twice, we could do with that second winning to help with our research. So if you do win it twice, you know who to call. The third, Peter McAfee, he got struck by lightning, and then won Tats Lotto, won the lottery, and odds of 1 in 2.6 trillion. So maybe 2 cancers in devils, the odds are very high, but perhaps it's just a pure coincidence. And the next person I want to talk about is Roy Sullivan. Before we get on to him, DFT1, first case 1996, DFT2, first case in 2014. Roy Sullivan was struck by lightning not once, not twice, but 7 times. Roy was actually a US park ranger, and maybe there was a reason. Maybe he was out in the field. So perhaps the devils are susceptible to transmissible cancers. There may be a particular reason, and our current research is aimed particularly to look to see whether devils are susceptible to transmissible cancers as well as our vaccine. So what are the implications for our vaccine work? We're developing a vaccine to protect against DFT1, but this new cancer, does that alter the scene substantially? Fortunately, DFT2 is similar to DFT1, and this picture shows the immune response against DFT1. So these are devils that are immunised with DFT1. Their antibodies in their serum react against the cells of DFT2. So the fortunate thing is that the cancers are similar, so protection against one may protect against both of them. 2 more names I'm going to mention are Edward Jenner and Frank Fenner. Most of you will probably have heard of Edward Jenner, and a lot of you would have heard of Frank Fenner. Edward Jenner discovered that dairymaids did not get smallpox. That's because they suffered from cowpox, which made them immune to smallpox. This he discovered 200 years ago. Frank Fenner is probably the father of microbiology in Australia, and he was a virologist, and he was charged with one of the responsibility to eliminate smallpox from the world. Can you imagine that? Eliminating the disease smallpox from the entire population throughout the world. And there is a statue in Geneva commemorating smallpox eradication. In 2010, it celebrated the 30th anniversary of smallpox eradication. So Jenner and Fenner, 200 years apart, one made a discovery to develop a vaccine for smallpox. The other led a trial whereby smallpox has been eliminated from the world's population. An absolutely monumental task. What have I brought that up for? Maybe it's possible that we can eliminate DFDD from the wild Tasmanian devil population. So the future of DFD vaccine research is we need to do a lot more research to improve the efficacy. An ultimate goal is to produce a single injection that will give lifelong protection. To do this, we may have to modify the DFD cells to be more antigenic so that we respond against both DFT1 and DFT2. We're analysing adjuvants to promote the immune response. Then we need to be able to deliver the vaccine. So this can either be the captive devils, the insurance population, prior to release, and then we will track those released devils, and there is a plan to do this in September of 2016. These released devils will have GPS collars so we can track them. Another goal perhaps is we could immunise wild devils, take a trapping trip, immunise wild devils and release them. If we can develop a vaccine that only requires one injection, we might be able to immunise devils in the wild and then release them and they'll be protected. So is it possible to eradicate DFDD from the wild? Jenna would have thought this was impossible. Fenner proved that it is possible. And I think we can do it. There's still a bit of work to do, but I think it's possible. So this picture represents a population of Tasmanian devils. The black and the grey represent Tasmanian devils. So what will happen is that a diseased devil, the blue devil, gets the disease and then bites a grey devil, transmit the disease, and then it bites another devil, and then bites another devil, so the disease is transmitted from one devil to the other, and it will continue, and those devils will die from the disease, which is the clear, those devils will die, and they will transmit the disease to other devils, and eventually the population will decline. If we look at the next slide, which says vaccinated herd immunity, if we can immunise and vaccinate enough devils, the red dots representing red devils, we may have a herd immune effect. So what may happen is that the diseased devils may bite a sensitive devil, transmit the disease, and die. But because around those diseased devils are all the vaccinated devils, the unprotected, the ones represented in black, are protected by the herd, so the diseased devils will die out, and with the diseased devil dying out, the disease dies out. All that we have left in this population are healthy devils. Those devils will then start to mate, as they do, and multiply, and the population will be re-established in the absence of disease. An ambitious aim? Impossible? Who knows? Jenner and Fenner showed that it was possible to remove smallpox. So why can't we remove DFDD in the island state of Tasmania? So to finish off and to summarise, devil facial tumour disease now represents two transmissible cancers. DFD1, originally called DFDD, was first discovered in the northeast of Tasmania in 1996. DFD2 was discovered in the Channel area a long way from the first cancer in 2014. Two similar but different cancers with similar mechanisms of transmission. We don't know how DFD2 escapes the immune system, but it's likely to be similar to DFD1. The transmission is a consequence of devils biting each other. DFD1 is a Schwann cell, arose in one cell in one female devil prior to 1996. DFD2, we don't know what the origin that is, and we're working to discover that. The devil has a competent immune system. It has a very good immune system. DFDD, particularly DFD1, escapes immune recognition by down-regulating MHC, so it is totally invisible to the devil's immune system. We know that if we immunise devils with DFDD cells, DFD1 cells, you can produce an immune response. We have discovered that if we treat the DFDD cells with interferon gamma, you upregulate the MHC, upregulate these immune recognition molecules, they produce a good immune response. Our current goal is to improve the vaccine to make the cells much more antigenic, produce a much stronger response, and eventually become a vaccine that just requires a single immunisation. We are undertaking field trials, and the ultimate goal is to eliminate devil-facial tumour disease from the wild. I believe this is possible. So to finish up, I'd just like to make some acknowledgements. The first, I need to acknowledge my wife, whose love and support has been important throughout the time as we try to work on devil-facial tumour disease. There's been some good times and some very depressing times working on this particular cancer. So there are ups and downs, and that moral support is critically important. Throughout the time, we've had a lot of people who have come through our laboratory who have played a major role, from PhD students to honours students to visitors, all playing some sort of particular role. And we've had devil keepers and vets involved. And that picture on the right is a photo of the lab, and I'll also make acknowledgement to John English, who sadly passed away in April of 2016, who was a devil ambassador. He took great interest in coming to see our research, and I just love him to know the progress that we are making. And the final acknowledgement slide just gives you an idea of the people we have at the work at the moment. We have a large number of people working in the laboratory at different aspects. We have people looking after devils, devil vets and the keepers. We also have a big collaboration throughout. We have CSL Limited. We have a linkage grant with CSL Limited. Elizabeth Murchison involved in the sequencing and helped us determine whether DFT2 was a new cancer. Kathy Belov and Wan Chen at the University of Sydney were very important for the MHC expression. Jim Kaufman and Hannah Siddle at the University of Cambridge are also involved in helping us understand how MHC can be re-expressed. Mena Jones and Rodrigo Hamidi within the field trials. Lynn Corcoran and James Murphy, Tony Pappenfuss at WeHi in our immunisation. So this project has started off with just myself and a PhD student, has become a worldwide collaborative progress. But this research couldn't happen without research funding, and pay particular acknowledgement to the Save the Tasmanian Program Appeal, who's without their support we would hardly be able to do research. They're through the Tasmanian, Dr. Eric Geil Tasmanian Devil Research Grants. We've had funding from the Australian Research Council, Mazda Foundation, University Foundation, Select Foundation, Morris Animal Foundation, Holsworth Wildlife Research Endowment Fund and Wildcare. So the research funding is critically important. So with that, I will finish, and I look forward to answering any questions that may be out there.
Thank you very much, Greg. We've received a question really asking about the vaccine, and is the ultimate goal to have one vaccine, which you've already addressed. So perhaps I can expand on the question posed to say, do you have a time frame? Time frames are questions that are always asked. No, we don't have a time frame. The sooner the better. Probably for the next 2 or 3 years, we'll start to refine it. Maybe 5 years would be a realistic goal to have a single injection. But don't hold me to that. And we have a question about whether the research was set back or how it was challenged when DFTD2 was identified.
Yeah, the research was set back a little bit because of this new cancer. But fortunately, this new cancer is very similar to the first cancer, so it really hasn't made a huge difference. The reason for this is that DFTD is transmitted from devil to devil, so it's just like another devil getting the cancer, and it's just more devils getting the cancer. It's not like it's a totally new phenomenon. So the research we've done in the past 10 years has allowed us to understand and detect this cancer, so that that background information has been critically important for us understanding this particular new cancer.
Well, thank you very much, Greg. I think that's we have no more questions being posed at the moment, but you've given us an enormous amount to think about with your really engaging presentation. So thank you very much for being part of the University of Tasmania first alumni webinar. I'd like to make note that this entire webinar has been recorded, and everybody who's been logged in today and anybody else will be sending you a link to the recording in the coming days. So once again, thank you very much, Professor Greg Woods.